Criminology Board Exam Reviewer. Forensic Chemistry. Definition of terms. Part 1. Absorbance. The measure of concentration of material present, the negative log, base 10, of transmittance of product of extinction coefficient, path length, and concentration, written as A equals IBC. Absorption. The incorporation of liquids or gases into the body. Absorption is also the process by which liquid hazardous materials are soaked up by sand, sawdust, or other material to limit the spread of contamination. It is a mechanical phenomenon wherein one substance penetrates into the inner structure of another, as in absorbent cotton or a sponge. It is an optical phenomenon wherein atoms or molecules block or attenuate the transmission of a beam of electromagnetic radiation. Absorption band. It is a region of the absorption spectrum in which the absorbance passes through a maximum point. Absorption elution. It is an improved, direct way of showing the presence of agglutinogens. In this method, antigenic material is first allowed to come in contact with antizera. The homologous antibody is specifically absorbed by a given agglutinogen. Absorption inhibition. It is a classical, indirect way of demonstrating the presence of an agglutinogen. This method involves the addition of a titred antiserum to the blood stain. Absorption spectrum. It is a plot, or other representation of absorbance, or any function of absorbance, against wavelength, or any function of wavelength. Absorptivity. It is the absorbance divided by the product of the sample path length. And it is the concentration of the absorbing substance. Acetaldehyde, Checho. It is a colorless liquid having a pungent and fruity odor, highly flammable and toxic, used chiefly to manufacture acetic acid. It is the first product of ethanol metabolism. It is also known as ethanol. Acetate. It is a salt or ester of acetic acid. It is a manufactured fiber in which the fiber-forming substance is called cellulose acetate. Where not less than 92% of the hydroxyl groups are acetylated, the term triacetate may be used as a generic description of the fiber. Acetone. The simplest ketone. It is a solvent for gunpowder. A highly flammable, water-soluble solvent. Acid phosphatase. It is an enzyme found in high concentration in seminal fluid. Any non-specific phosphatase requiring an acid medium for optimum activity. Acid phosphatase test. One of the most published and most widely employed techniques for semen identification. This enzyme can be found in the male prostate gland and is sometimes abbreviated as AP. Acid phosphatase in human seminal fluid originates from the prostate gland, often referred to as prostatic acid phosphatase. Acoustic coupler. It is a device used to attach a modem to the telephone system by placing the telephone handset on a set of rubber cups. Acrylamide monomer. It is produced by the reduction of acrylonitrile by either liquid ammonia or by calcium bisulfite. Acrylic fiber. Generic name for a manufactured fiber in which the fiber-forming substance is any long-chain synthetic polymer composed of at least 85% by weight of acrylonitrile units. Actinic rays. They are light rays of short wavelengths occurring in the violet and ultraviolet parts of the spectrum, which produce chemical changes, as in photography. Activation. It is the heating of the adsorbent layer on a plate to dry out the moisture and maximize its attraction and retention power. Active decay. It is that phase of corpse decomposition that follows bloat, is characterized by much maggot activity, and terminates with a rapid decrease in body weight. Activity node. An individual's past and present homes, current and previous work sites, and residences of partners, friends, and family members. Acute. Severe, usually crucial, often dangerous in which relatively rapid changes are occurring. Acute exposure runs a comparatively short course. Acute effect. A pathologic process caused by a single substantial exposure. Acute exposure. A single encounter to toxic concentrations of a hazardous material or multiple encounters over a short period of time, usually 24 hours. ADA, adenosine deaminase. An enzyme found in the serum of blood. Adaptation. The tendency of certain receptors to become less responsive or cease to respond to repeated or continued stimuli. Adenine. A nucleic acid consisting of a chemically linked sequence of subunits. Each subunit contains a nitrogenous base, a pentose sugar, and a phosphate group. One of the four building blocks of DNA. In the DNA molecule, adenine forms a chemical bond with thymine. Adenosine triphosphate, ATP. A nucleoside triphosphate that upon hydrolysis results in energy availability to processes such as muscle contraction and synthesis of macromolecules, including protein and carbohydrates. Adsorbent. The stationary phase for adsorption, thin layer chromatography. A solid or liquid that adsorbs other substances, for example, charcoal, silica, metals, water, and mercury. Adsorption. 1. The action of a body, such as charcoal, in condensing and holding a gas or soluble substance upon its surface. 2. The adherence of atoms, ions, or molecules of a gas or liquid to the surface of another substance. 3. Finely divided or microporous materials having a large active surface area are strong adsorbents. 4. The attraction between the surface atoms of a solid and an external molecule by intermolecular forces. Advanced chemiluminescent enhancement system. Used for the non-radioactive quantitation of small amounts of human DNA. AGAR. A polysaccharide extracted from seaweed. A gelatinous product extracted from certain red algae used chiefly as a gelling agent in culture media. Agarose. The neutral gelling fraction of agar commonly used in gel electrophoresis. Agglutination. The clumping together of living cells as a result of a reaction between the cells and an appropriate immune serum. Agglutinin. A chemical product of the process of immunization rising in blood serum and causing the red corpuscles, with which it is brought into contact, to coalesce into floccules. Agglutinogen. An antigen that stimulates production of a specific antibody, agglutinin, when introduced into a host animal body. Outdated term for red cell antigen. Aerosol. An outsole or midsole incorporating an air pocket or cushion. Albumin. One of a group of heat-coagulable, water-soluble proteins occurring in egg white, blood serum, milk, and many animal and vegetable tissues. Alcohol. An organic compound having a hydroxyl group attached. The lower molecular weight alcohols, methanol, ethanol, and propanol, are water-soluble. Alcohol dehydrogenase, ADH. The main enzyme that catalyzes the conversion of ethanol to acetaldehyde. Aldehyde dehydrogenase, ALDH. The enzyme that converts acetaldehyde to acetate. Algorithm. A set of well-defined rules for the solution of a problem in a finite number of steps. Aliphatic. One of the main groups of hydrocarbons characterized by the straighter branch chain arrangement of constituent atoms. Aliphatic hydrocarbons belong to one of three subgroups: 1. Alkanes or paraffins, all of which are saturated and comparatively unreactive. 2. The alkanes are alkydians that are unsaturated and are more reactive. 3. Alkenes, such as acetylene. Aliquot. A measured amount of liquid taken from the main portion. Alkali. A basic substance, pH greater than 7, that has the capacity to neutralize an acid and form a salt. Alkaline phosphatase. A phosphatase active in alkaline media. Alkaloid. One of a group of nitrogenous organic bases, especially one of vegetable origin, having a powerful toxic effect on animals and man, such as nicotine, cocaine, strychnine, or morphine. alkanes. An aliphatic hydrocarbon having the chemical formula C6H2N+2. Also known as paraffin. Alkyd resin. A class of adhesive resins made from unsaturated acids and glycerol, used as a bonding agent in paint and lacquers. Allele. One of a series of alternative form of a gene, or VNTR, at a specific locus in a genome. In DNA analysis, the term allele is commonly extended to include DNA fragments of variable length and sequence that may have no known transcriptional product but are detected in a polymorphic system. Allele frequency. A measure of the commonness of an allele in a population, the proportion of all alleles of that gene in the population that is of this specific type. Allelic marker. Allele form of a gene used to identify chromosomal segments suspected of association with a certain phenotype. For example, allelic markers may be used with a family pedigree in which a phenotype is common to identify chromosomal segments that contain the gene responsible for the phenotype.